other busy schedule and other things, one might have not been ready for the presentation. So it is flexible. Those who are ready with your presentations, you can present and interact. Second thing is, during the address, our director has told for to make an arrangement for field visit. So it is planned for the second day. So second day afternoon, that is tomorrow afternoon, we will make an arrangement for visiting the campus, main campus, and also there is a separate department on organic agriculture. So where also we can go and interact. They have their own fields, so we can have an interaction with them. And the third one is that, so all the delegates, those who are traveled by air or by train, kindly give your TA bills. That has to be, because we have to submit to office to check for the tariffs and other things. So then only it will be processed immediately. So this is for your kind attention. The TA bill will be given by Dr. Anand. So you can fill your details and submit back to him. So he will discuss with the office people and he will revert back to you. If they say, is there any discrepancy, he will revert back to you. So these 3 points, so I want to emphasize. So now we can start the technical session. I think everybody is here. Shall we start? Okay, so we will break around somewhere around 1:30 for the lunch. My humble request is that as per the plan, it was 30 minutes for the presentation, but suddenly we have to accommodate the field visit for half day tomorrow. So my request is that if possible, try to complete by 20 minutes. That will save some time, then we can accommodate all the presentations. Thank you.
Respected delegates and participants, we shall now formally commence the first session of the seminar, chaired by Dr. P. Nallathambi, ICAR IARI Research Station, Wellington, and Dr. Sika Arbino, Montpellier-sur-Lez, France. We now welcome our co-chairpersons, session chairpersons.
Good morning to all. Again, I thank these organizers for giving this opportunity. We are great privilege to have the three speakers. First, I invite Dr. Andy Kumar. He will present on the plant microbes for the sustainable agriculture and food security. To brief about Dr. Andy Kumar, I think all pathologists and in fact the plant protection scientists know that he is an eminent plant pathologist and currently working on the microbiome. And interestingly, most of his students are well placed in the system, ICR as well as the other organizations, because of the quality of the work and the publications he contributed through his students. I think it's a brief, the contributions are already in the abstract book, you can go through it. So without taking much time, as told by Dr. Angappan, I request Dr. Kumar to restrict to the 20 minutes so that we'll have some time for the interaction. Thank you.
So at the outset, let me thank the organizers for giving me an opportunity to share whatever we have been doing in IRI, that is Indian Agriculture Research Institute in New Delhi, and my own thought process about the subject matter, what I'm going to deal today, that's plant microbiome. This subject is not new, but I would say the new concepts that landed in the genomic era is taking us to different areas of agriculture research. About my team, we have been working on this plant microbiome for the last 14 years, and we gained some experience, not all, but little experience, and those experiences really gave us some kind of confidence how best this plant microbiome can contribute to sustainable agriculture and food security. So myself, a professor in plant pathology in Indian Agriculture Research Institute in New Delhi. Few slides, I will take you to the tour of the realities in life. That's all about population versus food supply. This has been always a challenge before all of us. In the last 300 years, I think all of you must be knowing Malthusian's theory. And every time we produce more to feed the entire population. So this is a snapshot of what would be the population in 2050. We are going to cross around 9 billion people. So the global population is going to cross 9 billion. So that is a projection. So you just pay attention to this green one. Most of the population is going to live in developing countries, where food production is always a challenge. So World Bank has launched 4 different aspects to meet this challenge. And those aspects are very straightforward. Innovations, enabling market, supporting people, and building political leadership that also take the regulatory regime on board. So the first one is innovation. So inspiring innovation is going to really contribute to sustain the production, to keep pace with the population explosion. Innovation encompasses everything. Every timeline in the history of the modern time, innovations really brought us to new area, and we always succeeded in our life. Inspiring innovation also need new thought process and a new line of thinking, and of course, technology need to be integrated, and aligning with the new thought process and technology, certainly we can meet the objective of sustainable agriculture and the so-called food security. This is a reality. 20 years back, we never talked about food waste. Now the food waste is considered as one of the major factors in the world to meet the hungry human population. This is data what I could gather from the literature. The pre-harvest losses are around 5 to 20%. We could bring down to some extent thanks to the chemicals and the modern agricultural practices that came into practice from 1950s and 60s. Post-harvest is still bigger challenge. We are unable to really reduce the losses, especially on fruits and vegetables. Need a different kind of thinking. Food waste is the new in the whole, losses, which otherwise would have fed the hungry human population. This is a report, a 2024 report. I just borrowed from UNEP. If you are interested, please go through it. Alarming pictures. They say you don't have to produce more, you try to reduce the loss. That itself is sufficient to feed the population what we are going to experience in 2050. So in terms of money, roughly 1.3 billion tons per, in terms of volume, 1.3 billion tons is lost or wasted. So this is roughly equivalent to around $1 trillion in economy terms. This is a ground reality. Whether we accept it or not, yes, this is happening. I think all of you must have experienced wherever you go. I would say in India, the marriage halls are living examples where how the food is really wasted. And this is alarming. And come to specific crops. That's also very, very important. I think the entire human what do you call calorific needs are met by 4 major crops. I think all of you must be knowing, starting from rice in Asia, wheat in Europe, corn and potatoes in South America and Latin American countries. And these crops are the major. And of course, you see the loss is what is recently observed in these crops. And it's a huge loss. And the challenges. Challenges are, this is a recent paper in APS. The challenges, which is addressing a small fraction of the losses caused by plant diseases. What should be our strategy to reduce those losses, right? And those who are interested, it's an open access paper. You can read this paper, Key Challenges in Plant Pathology in the Next Decade. And I really enjoyed reading this paper. And they also narrated around multiple points, I would say narratives, to reduce those losses. And among them, I just picked few of them. 2 of them are entirely on plant pathology. Mechanisms, epidemiology, management of insect-transmitted plant pathogens, mostly the viruses. And of course, another point is the microbiomes. Microbiome has emerged as one of the next big subject which can lead us to develop next generation crop protection technologies. And this is highlighted as one of the next big thing in agriculture. With this background, I would say that I would take you to the fundamentals of plant microbiome, where it all started, and how it is progressing, and what it can deliver in the long run. So this is my objective of the whole lecture. So historical background, I think, let us not forget, the subject plant pathology and microbiology, I would always say they emerged out of tragedies. The late blight outbreak in Ireland and Europe that brought us a new science in agriculture, that is plant pathology, and the associated developments. We buried spontaneous generation of disease theory, germ theory emerged. All this happened in a close succession from 1860 onwards. And I will be touching upon the key players who did all those fundamental work, the new concepts, why microbiome matters, and how this microbiome can influence the disease framework. That's going to be my second major chunk of my presentation is on that. In sustainable agriculture, how the microbiome approach can lead us to develop a next generation crop protection technologies. And of course, Government of India, I'm sure Government of France also might be promoting these agri-biologicals as a supplementary technology to sustain agriculture production. How this microbiome research can lead us to develop new agri-biologicals which can be used parallel to the chemicals, if not to eliminate the chemicals, because the chemicals are so important to sustain the production. At the max, we can reduce their role or their input, supplement with the microbiome-based technologies. That's going to be a few slides on that. Maybe the take-home message and the discussion on that would be my last slides. I already told subject pathology and microbiology emerged out of tragedies. Of course, that tragedy led us to a new science. This is some of the timeline what I thought is pertinent to this audience. Microscopes were invented in 17th century, 18th century. 19th century, we saw germ theory coming up, pure culture techniques. People like Anton de Barry, we consider him as a father of plant pathology, Louis Pasteur, Robert Koch, all of them contributed for the foundation. The entire theory is sitting on a simple principle, what is called reductionist strategy. So reductionist strategy is very important to prove the cause and effect relationship. It was needed at that time because to prove what is really responsible for the disease, not only plant, but also human and animal diseases as well. And 90s, I think, all of 1900 onwards, the 20th century, all of you must have studied the different methods of classification, technology integration in science, new way of looking at the microbes, ribosomal or RNA as a marker for identifying the diversity. We could see the development eventually graduated into what is called as genomic era, started from 1995, with the onset of or the launch of the first genome sequencing of Haemophilus influenza. That set the ball rolling for genomic era. Genomic era also witnessed the microbiome research emerging in the rapid pace. I would say the subject microbiome really evolved in a geometric proposition from 1995 onwards. So 2010 onwards, we started integrating different omic tools to understand the role of microbes, not only the role, the taxa of microbes, which we could not really understand through the conventional methods. So focusing on transcriptomics, metabolomics, you put any omics, it is nicely fitting here. And of course, the system biology people started looking at. And presently, of course, we are talking about microbiome engineering, application in health, the so-called One Health, agriculture, environment, and whatnot. So they are the main players. I just thought of, I think many of them are from France as well. So they played a very important role to shape the science, pathology, and microbiology. So come to the flow of technological development, conceptual, what do you call, understanding of the subject. We started with isolation based on the reductionist strategy. Pure cultures are so important. All of us are really bothered about pure cultures even today. Germ theory, Koch postulates to prove the cause and effect relationship, causal agents. And now the sum total is leading us to a new era, what is called as the system biology strategy, to understand the system as a whole rather than in piece by piece. So that is the so-called the current concepts. So for the youngsters who are sitting here, or those who are not too familiar with the subject, terminologies are very important. Microbes are not equal to microbiota. Microbiota is not equal to microbiome. So there is no longer microflora. So these are all some of the conceptual changes as happened in the recent times. I call upon all of you to read this particular paper. Microbiome appeared in Microbiome by a large number of authors. To make the long story short, microbes, when I say it is an individual pure culture, and when I say microbiota, it's a community of species. When I say it is a microbiome, it is a microbiota plus a theater of activities. So microbiome is not a solitary individual species, rather the functions integrated in the system. It's not that easy to study microbiome, I must tell here. You can study microbes, you can study microbiota, but to study the microbiome, you need a different kind of tools and techniques, because microbial interactions are very difficult to study. The end point of one microbial metabolism is starting point, the constant flux of changes. So this is the reality. So first time he coined the term microbiome. Disease. But you are not going to see the disease epidemics. To get the disease epidemics, the time frame comes as another factor. But I am trying to fit in here where exactly the microbiome can play a role here. In my perception, the microbiome can be the fourth dimension we ignored all these years, because the microbiome can influence each and every factor of the disease triangle. Let me take you the tour of few slides, how this microbiome, the theater of microbial activities or theater of microbiota activities, can influence each and every disease triangle factors. When I talk about microbiome, it's a broad term. I'll just take you to plant microbiome associated with the plants only. And within the plants also there are multiple niches, and those niches are completely contrasting many times. The below ground niche is different from above ground. contrasting. You won't find any similarity. It's a fairly insulated below ground and highly variable above ground. There is a sunlight in the above ground, completely absent in the below ground. Is aquatic ecosystem in the below ground, the terrestrial ecosystem in the above ground. These two are completely contrasting, and the microbes what is living there is also completely contrasting, right? So these microbiome, whether it is a rhizosphere, phylosphere, or endosphere, or any sphere you talk about plants, they have their own say in the disease triangle. Let us see what it is. Microbiome as a fourth dimension in the classical framework. The plant-associated microbiome, which includes the different sphere-associated microbiome, how it can influence a virulent pathogen. Let's see how it influences. So the virulent pathogen is one among the microbial community which is habitating on the plant pot, right? It is one among. It's a minority. The majority of them are the whole lot of millions or trillions of organisms which is living there. It's a minority. But still, it is able to cause disease. These are all some of our thought process, the ways and means of the microbiome influencing the pathogen or the pathogen associated the roles. I think all of us must have studied in biological control. We talk about competing microbiomes or competing organisms. It's a microbial competition which is going on. In our own experience, we increase the number of certain beneficial organisms in the particular sphere, let's say a foliar microbiome, you start seeing a remarkable reduction in the particular pathogen which is also colonizing on the foliar part. So this is simply by number, competition. So we also see many times very close collaboration between two different organisms, Pentoias and Xanthomonas, Enterobacter and Ralstonia, and so on and so forth. There are many such examples are there. And my pathogens can be predated by the microbiome-associated organisms. That is another factor. And attenuation of virulence is a new thought process. Pathogen can be there. It's not getting killed, but its quality is changed by microbiome components, what I call virulence modulation by microbiome. And of course, somebody may talk about pathobiome today. Pathobiome disruption of microbiomes is the other way around. Pathogens can disturb the microbiome as well. So we recently published a paper, the dysbiosis associated microbiome, how the pathogen disturb the microbial balance on the plant pot. So it's a very recent paper from our group. So what I'm trying to say here is pathogens are under the influence of microbiome components, and if you modulate the microbiome on the particular niche, you are ready to manage that particular pathogen caused by causing some disease. That's our thought process. And these are all some of the snapshot of our own microbial candidates, what all it does. And I put under the two major category of the metabolome, secreted as well as volatile. We could see all kind of permutation combinations. Some are producing secreted metabolome, some are volatile, some are both. It's a whole lot of dynamic system which is operating there. So this is going to happen when pathogens are interacting with microbiome, other way around, the microbiomes are interacting with pathogens. So that was all about how microbiome can modulate pathogen behavior and pathogen numbers. I'll take you to how the microbiomes can influence the host. This is one of the hot topic. Now everybody is talking about plant immunity and how microbiome can contribute or enhance the plant immunity by meddling with the plant gene expression.
Sorry, Dr. Kumar, I should interfere.
Yeah.
Your time is another 5 minutes.
No problem, we'll take. So host and host factors, especially the susceptible host, is under the influence of microbiome, and these are all some of the ways and means the microbiome can influence the host. For example, immune modulation, nutrient acquisition, growth promotion, stress tolerance, Yeah, it's a very interesting question. If you talk about the microbiome, I already told everybody is doing, but where it is going to lead us, that's very important. So our protocol is we need to identify the core microbiome of that particular plant species. And what we found was the core microbiome is invariably something is doing a lot of benefits to the plant. Core microbiome is the one which is inherited in different generations of that particular plant. For example, Pantoias. Pantoia is inherited. The question is how it is inherited, this big question mark, but you plant a particular genotype in two different agroclimatic zones in the country. For example, Himalayan zone and Port Blair in Andaman, you start seeing the same genotype of microbes in that particular plant in the phylosphere. Exactly same, it's a kind of twins. So how it landed there? It is inherited through the seed. It may be seed transmitted. The point here is such an organism is, in our opinion, 60 to 70% of the core microbiome associated organisms are always contributing to the plant survival, plant ecosystem function, disease suppression. I would say that if you are able to come out with the core microbial members, you can use them as an agribiological active principle. And they are not a negative organisms at all. Yeah, not at all. Because the negative organisms, whatever you were talking, they are coming under the category of, to our knowledge, satellite microbial members. Because it jumped from one system to another system. It never got inherited with the plant. Right? For example, we normally encounter some of the notorious organisms like Staphylococcus aureus. You see on leaf, we eliminate on the first instance itself because it's from human skin, it might have gone to the leaf because handling matters a lot, right? And also you start seeing some of the very promiscuous organisms like Pseudomonas aeruginosa that is being promoted as a biocontrol in many, but you will never be able to commercialize it. Mark my words, because it's a notorious organisms, and what we consider as a biocontrol trait is actually a virulence factor for medical people. Yeah.
So, yeah, please.
I would like to ask that the microbiome largely, it's mostly conservative sometimes, and sometimes it is, as you mentioned, surviving its own way. These two approaches we are following, really is now mechanized world and highly input agriculture, intensive agriculture. How do you see that really the technology can boost to balance both intensive agriculture and as well as conservative agriculture?
Exactly, yeah. We did have some experience on that, comparison between the two systems of agriculture. What we found was in a short time span, we could not find any signature microbes associated with the conservative or modern agriculture. But the point is not that. The point here is, in modern agricultural crop, monocropping and narrow genetic base, these two are the real culprit. And we need them actually to sustain the homogeneous production of any agricultural produce. Everyone wants the same size tomato, same color tomato, right? That is what? It is actually a homogenization in the end product because the genotype is narrow. And if you ask me, the so-called pathology evolved from such a scenario only. There is Irish potato landed in Europe. They promoted it. It's a monoculture, narrow genetic base. The microbiome, which otherwise would have colonized the wild ancestors, are missing here. In the process of breeding, in the process of selection, we lost all those microbes. What we call, we domesticated crop domesticated few microbes, or microbiome. And those microbiome pave the way for the emergence or re-emergence of plant pathogens. If you bring those microbial candidates, which was there in the previous ancestral plant, there may be some solution. The question what you were asking, if you bring them in the agricultural practice, maybe we can reduce the chemical input to some extent, because microbes have evolved with the plant and they play some, it's a kind of give and take. They give something to the plant, in return they get the shelter in the plant. Certainly, we need to identify what is the factor which the plant take from microbes, which can help the agriculture or whatever One Health we are talking here. Yeah.
I think Dr. Kumar will be available for another 3 days here. I think we can allow one more question because we have another 5 speakers.
Just, yeah.
Kindly restrict to their questions. Okay. And you can have the free interaction in the other time. We can allow one more question.
Thank you, sir. Thanks, Andy. Excellent presentation. I always love to hear your PPTs. So the question is actually in the last slide you mentioned about microbiome-based products. So the last step. So when it is about microbiome-based product, it is about consortium of organisms that we are going to apply, or with different functions, or when we apply, that is going to alter the microbial community structure in the soil.
There are two strategies, with the permission of the chair. It's a very important topic. Why? Because regulatory regime in our country doesn't allow consortium. Point number one. Right? If you want to take microbiome research to the commercial domain, there must be some kind of alteration in the regulatory regime. They should allow the core microbiome members in the formulation. We have to justify that core microbiome are integral part of this extended plant genome. We always see. So that means what? At least, if not many, at least a few core microbiome should coexist in the same product. Then the objective of microbiome can be fulfilled. That is one way of doing it. Another way of doing is you try to put the precursors in the rhizosphere or phyllosphere, encourage the native microbiome, the core microbiome, to flare up their population. That is another strategy is emerging now. You go for precursor application on the plant or plant associated niche, that is going to do the job. You don't have to apply the microbiome at all. Rather, you promote few select microbial members in the core microbiome to come up in their numbers, or the theater of activity is activated there by the precursors, then the job is done. End of the day, it is a chemical ecology which is going to do the job, whether it is microbe live cell or whatever. It's a chemical ecology is going to contribute for the plant survival. Thank you.
So with this, we thank Dr. Kumar for your wonderful interaction. I think others can freely interact with him. With the permission, can we invite the next speaker, Dr. Chen, to speak about the worldwide diversity and ecology of seed-borne bacterial phytopathogens of bean. I think all the speakers, the significant contributions are already in the abstract. Please. Dr. Chen, you have to restrict to the 20 minutes. We'll keep some time for the
presentation is not there.
So don't count this time.
Yeah, I will keep the track of time as well. Try. Yeah, I prepared for 30 minutes. I will try to rush, but not rush too much. So, okay. Is it starting or? So, hello everyone. Thank you very much for the invitation. I will do a talk that will be much more focused now after this talk about the microbiome, and I will focus on bean. Don't pay the title. I wrote it a 4 genetic lineages that we call GL1, 2, 3, and fuscans, belonging to 2 different species, Xanthomonas citri patovra fuscans and Xanthomonas faszeoli patovra faszeoli. So the faszeoli patovra faszeoli is quite distant phylogenetically from the others. So this is what we call convergence, pathological convergence. And if we now try to go more global about this bacteria, the disease is worldwide, globally widespread. Okay, so everywhere you grow bean, you have this disease, even in India here. Okay, but so we analyzed a broader diversity of this pathogen using sequencing of the GRB housekeeping gene, and we could distinguish between these 4 lineages that I showed you before. And as you can see, the distribution of these lineages is different in the different geographical areas of the world. You can see that in many countries, the genetic lineage fuscans is more prevalent than the others. But for example, in the US, that is the GL1, that is the one that is phylogenetically distant from the others, that is more prevalent. And in some places, like the small island of La Réunion, you can find the 4 genetic lineages even in the same field. So maybe they can exchange genetic materials, and it will be more maybe difficult to build genetic resistances about this. And what you can see maybe that is missing here is that in this part of the world we don't have so much results on information about the diversity, and especially in India. So that's one part of the thing, that's why I'm here. If you want, maybe we can discuss about this later. Okay. So this is about the diversity, and now back to the convergence. I showed you already these 3, and we made comparative genomics analysis, and we pointed out that there were a lot of horizontal gene transfer that we believe are responsible for this pathological convergence. We found more than 100 genes that were 100% conserved between the green one and the yellow one, so we believe that these genes were horizontally transferred from this one to here, and that they are responsible for the acquisition of the capacity of the faszeoli to infect bean. Okay. Among these 100 genes, there are 4 particular genes that are called X-fetal 1, 2, 3, and 4, and all these 4 genes, they belong to the TAL, the genes that are encoding TAL protein family. Okay, so you can see here that X-fetal 1 is shared between the 4 lineages, so that means that it was part of the horizontal transfer that is here, and also X-fetal 2 was horizontally transferred between the fuscans and GL1. This was not the case of the X-fetal 3 and 4. Okay. So why are these TAL effectors important and for us? Because they have really, their activities, their functionality is quite specific. So TAL is for transcription activator-like effectors. In fact, these are proteins that are really act like eukaryotic transcription factors. So when the bacteria enters the plant tissues, it comes in contact with the plant cell, and the bacteria, I think maybe a lot of you know about this, but the bacteria employ a type 3 secretion system to inject in the plant cell a lot of type 3 effectors that are small proteins that can maybe disrupt the immunity or favorize the aggressiveness of the pathogen. And the TAL effectors, they are really specific. They migrate into the plant nucleus. Then they are able to bind to DNA in a specific sequence of DNA that is usually positioned in the promoter of susceptibility genes. And the TAL effectors is able then to recruit the transcriptional machinery and induce the overexpression of the genes downstream. So this is a way for the bacteria really to manipulate the transcriptome of the plants so that the plant induces the production of transcripts that are beneficial for the pathogen, and you have more disease. And how, so this is one part of the thing that I wanted to say, but also what is important for us is that the TAL effectors, maybe you know about the ETI effector triggered immunity. they can be recognized by ETI, but also the plant developed a very specific kind of resistance against these tals. For example, the most obvious, the most simple for the plant is to mutate the sequence that is targeted by the tal. That way the tal can't bind anymore and the susceptibility gene is not overexpressed anymore. So here you have the mutation of the target can bring to resistance by loss of susceptibility. That means that the bacteria is less aggressive because the tal is not functioning. Another way for the plant to specifically resist is that the target can be relocated, maybe by segmental duplication, in front of a gene that is not a susceptibility gene, but that is a resistance gene. So this time the bacteria, when it employs the tal effectors, then it will induce a resistance gene instead of susceptibility gene. So the gene transcript will bring resistance, and the bacteria will not kill itself, but it could be viewed as is. So that is why we call this gene executor genes. So with these two examples, you can see that in fact the research of tal effectors can facilitate the search for new resistance gene on new loci to develop resistances. And one important part is that the people that are working with tals, they developed bioinformatic tools to predict using the sequence of the tal and the sequence of the whole genome of the plant. We can predict where the tal will land. The bacteria have different tals that target different parts of the genomes, and we have bioinformatic tools to predict this. Okay, and another thing that can be useful with this tal is that there is a possibility to engineer artificially the tal effectors. Maybe some of you know about the TALEN. The TALEN were developed from tals with binding with an endonuclease for genome editing. So, but CRISPR-Cas was more efficient, so the TALEN were now maybe not used anymore. But also you can do something even more simple. You can build a designer tal that is just an artificial tal that will overexpress the candidate genes, but contrarily to the natural tals, this one you choose where it lands in the plant genome. So you can induce any candidate gene you want. For example, you can induce a candidate resistance gene, and that's what we did with BEAN. We published this early in 2025. We used this approach to induce a gene that was a candidate air gene, and we proved that it was efficient for symptom reduction. Here, I will not go into details for this particular result, but just this result is that here is the bacterial control, and here are 3 strains expressing different details that are all targeting the promoter of this PV-OFP7 gene, and you can see that you have more than 60% symptom reduction, and this symptom reduction is correlated with the level of transcription of the gene. Okay, so this is the end of the first part of my talk, 11 minutes, so it's okay. So the first take-home message is that common bacterial blight of bean is due to diverse xanthomonas species or lineages, that convergence toward infecting bean is due to horizontal gene transfer that include tal effectors, and these tal effectors can be used to accelerate the discovery of new resistance genes. So that's part of the first thing that I wanted to say. The second part I told you about is the seed transmission. And all the slides that I will present now is not my particular work, it's more the work of Armel Darras, who is a researcher in our group, and she is really specialist of seed transmission. So a first result here is that she injected inoculated the bean flower buds using, here you have a pipette tip, a yellow pipette tip, you have here a droplet of inoculum. You can inoculate directly the bacteria inside of the flower bud and see if, so this is the floral pathway. You wait for 6 weeks, you take the seeds, and then you macerate the seeds, you plate, and you get the bacteria. And you can see that with this kind of inoculation, you have more than 80 to 90% of seeds that are infected by the bacteria. Another thing that she did is that she inoculated by spray inoculation, and as you can see here, the flower buds, she covered them with plastic bags to protect them. So the flowers were not touched by the inoculum. That means that the bacteria had to enter the leaf, go to the vessels, and then to reach the flower buds, and then go to the seeds, okay? And when you do this, you still have a transmission of the bacteria, but less than with direct inoculation. You go to up to 60% of seeds that are infected. And she did this not only with the wild type strain, but also with strains that can't express the type 3 secretion system that I showed you before. And when you do this, you lose completely the ability to go to the vascular pathway, and you reduce a lot the transmission of the bacteria, even if you inoculate them directly in the flower buds. So that means that the type 3 secretion system is really important for the bacteria to be transmitted. And so that means also that the seed transmission it is really an active process, and the bacteria is interacting molecularly with the plant cells. Okay. So we wanted to go further with this analysis of molecular interaction, and in collaboration with another researcher from IRHS, that is Jérôme Verdier. And here is what we did. So this is Armel, again, that did this. So here you inoculate the plant as the same with spray inoculation, but without putting the plastic bags on the buds, so you have really a big inoculation of the mother plant. And with other plants, you inoculate water as control instead of bacteria. And 24 days after pollination, you take the seeds are still small, that are really small green seeds, immature seeds. And so you remove the seeds from the pods and you take them. Then 30 days after pollination, and then 40 days after pollination, here you have really mature seeds that you could plant and grow new plants with them. And with all these different batch of seeds, we perform dual transcriptomics. That means that we took the RNA-seq from the plant and from the bacteria, and we compared the the seeds inoculated with bacteria compared to the control. So what we got here is that, so here it's at 24 days after pollination, 35, 42 days after pollination. You can see here that is the transcriptome of the plant, that early the plant is really impacted by the bacteria. A lot of genes are upregulated or downregulated, and among the genes that are upregulated, there are LRR genes involved in perception, and among the downregulated, there's the response to stress, like for example, HSPs or MAP kinases, they are downregulated. So we believe that the plant is still able to sense that the bacteria is there, but maybe as we see that also the bacteria is expressing the type 3 secretion system, maybe the type 3 effectors can reduce the stress responses and make the plant susceptible. But what is important to see here is that here you have a decrease in the response of the plant during the timeline. At the end, you have almost no response. That is maybe obvious because the seed is maturating and is entering dormancy. Okay, but what was very interesting for us is that the bacteria is doing the same. It's as if the bacteria is also going in dormancy, and we believe that is an adaptive process for the bacteria to not impact too much the seed and to be transmitted to the next generation of seeds. Okay. We call this the ceasefire model. Okay. Maybe if I have maybe 3 minutes, I will go to the two last slides of my talk. This is not the end of the story. Here is the same thing that I showed you before, but then we took the mature seeds and we imbibate these seeds again, either with water or with the bacteria here, and here with the bacteria. So here you have water plus water, water plus bacteria, and bacteria plus bacteria. And we perform qPCR on defense genes after 3 hours of inhibition. And what we got was really interesting. Some of the genes that we targeted, they didn't have any response. Here you have the water plus water, that is the control. And here the mother plant is inoculated with water than bacteria. You don't have any difference, and bacteria, same. But here, these two particular genes that are really interesting, because when you inoculate the mother plants with water, then the seeds with the bacteria, you have an increase in the expression of these genes. So that suggests that this gene is able to sense the new bacteria that entering the seeds. But when you inoculate the mother plant with the bacteria first, it's the opposite. That suggests that the bacteria here that were infecting the mother plant, they silenced these genes for the next generation of plant. And we believe that with this silencing, it helps, it makes the seeds, the next generation of seeds, more susceptible to the disease. Okay. So the take-home message too is that seed transmission of the Xanthomonas is an active process, and that during seed transmission, Xanthomonas is able to lower its aggressiveness during the time, and also to silence putative defense genes. And we believe that this could help the bacteria to colonize the next generation of genes. So with this, I would like to thank all the people that were involved in this work, the different facilities and the fundings, and you for your attention and the CEFIPRA for funding this meeting. Thank you.
Thank you, Dr. Chen. You very, very smartly you managed your time by showing that but you exceeded 3 minutes. Anyway, we'll allow for 2 questions.
Yes.
Number one, you talked about loss of susceptibility gene. And I believe loss of susceptibility gene expression, right?
Yes,
Does it have
yes, but it brings that the plant is more susceptible.
Yeah, the loss of
is more resistant. Yes, because
the loss of susceptibility expression.
Yes, expression.
Does it have any fitness cost for the plant? Because we all believe that susceptibility genes are equally important for the plant development.
Oh, I don't know about this. The susceptibility genes, usually, for example, I will go back here. The one of the most studied susceptibility genes for the infected, for the targeted vitals in rice are sweet genes that are genes that are encode sugar transporter. And so we believe that the sugar is exported to the apoplast, and that maybe the bacteria feed about this. But I don't know about the fitness cost.
This is one of the questions normally being asked now. Can we touch this susceptibility gene for genome editing projects? So then this question comes, it may have fitness costs for the plant. So anyway, so that's, we don't have answers for that.
But as you target the promoter, it shouldn't impact too much.
Yeah, yes. Yeah. My second question is about horizontal gene transfer you talked about.
Yes.
What pathway? Is a conjugation or transformation or is a cage mediated? Which horizontal gene transfer you have some idea about that?
Among the different, the question is about the horizontal gene transfer, these genes. So a lot of the genes that were involved in this horizontal gene transfer are encode like hypothetical proteins, but a lot of them also encode the type 4 secretion system that is involved in conjugation. So we believe that it's
that's fine. But what is some actual mechanism by which the HGT transfer?
We don't know. We believe that maybe it could be the conjugation. Yes, conjugation. Yeah.
Thank you so much.
Yes. You're welcome.
One more question. Please be very brief in your questions so that we will save some time.
That's it. There is one question still.
Yes.
I'm just wondering whether you have TALs inducing susceptibility genes and TALs inducing resistance genes. Do you have obvious differences in between effectors from both categories? Or not at all? Or sometimes do you have some effectors able to do both, induce resistance and susceptibility?
Yes, in fact, it's kind of obvious that the bacteria developed genes to target susceptibility genes, but in different plants, when you study the diversity of the plant, you find few genotypes that are resistant. So they develop this resistance against the TAL, but this is the same TAL that is this is the same TAL.
Okay, thank you, Dr. Chen, for your nice presentation. Next, we'll invite Dr. Valarmadi on cotton necrosis. Yeah, please. 12:40. Exact by 1 o'clock you have to close.
So, good afternoon and all, and welcome to my presentation on the necrosis disease of cotton, which is an emerging one, and how to manage it. So this is the institute where I belong to. It is an ICR, Central Institute for Cotton Research Regional Station, which is located near to the Agriculture University. And our headquarters is at Nagpur, and we have another regional station which is at Haryana, which is Sirsa. And when we speak about cotton, so it is a non-edible crop. And if you see, 77 countries has been cultivating cotton, of which 105 countries has been consuming as for its textile industries. So of the world, you see the top 4 producers are the United States, Brazil, and China, and India is also one of the top producers. So when you speak in India, we usually refer cotton as a white gold, and it is a cash crop. So it is a leading producer of which is competing China, and by 60%, 5% of cotton fiber which is used in textile industry. So this slide I have given here is our target in 2030 was to increase our productivity as also production to 8.95 million tons, where at present we have 6 million tons. So when you see, the productivity is very less when you compare to the global scenario. So we have only 450 kg length per hectare. We have to increase up to 746 kg. So for that, in order to meet our demand, like a 47 meter cloth per capita. This is the target. So this is the means the area of production yield of in India. So when you see, there are the 4 species which we cultivate in India. The diploid cotton, which is Gossypium arboreum and Herbaceum. Gossypium is a tree cotton where 2% contributes, and this cotton is mainly we use for the surgical purposes. And this Herbaceum is only in the research study, and this we not found in the farmer's field. The other is the tetraploid cotton, which is Hirsutum and the barbadense. So when you see, this is the American upland cotton, which contributes 90% of our cotton, and where this is the BT cotton where it is derived. And the other is the barbadense, with the origin is from Egypt. We call this as a extra long staple cotton also. And when you see what are the challenges, when you speak about why the cotton productivity is low, when you see all those years in India, when you compare to the other countries, is narrow downing of our genetic diversity was the first reason. And the other is the one we developed BT cotton in 2003. It is mainly for the American bollworm. But those BT cotton are now said to be resistant to the pink bollworms. This is the recent scenario. And the other one is we are expanding our long duration hybrids to the marginal areas also where it is not suitable. And also the increased frequency of extreme weather events, and also soil health we are not concentrating. The other most important is the pest and disease outbreak in cotton. And also the deprived technologies what we are developing by the researchers or the scientists, it may not be reach up to the cotton farmers. So when you see about what are the diseases in cotton, so we can see the soil-borne disease like root rot, wilt, and the fungal foliar and bacterial foliar disease also there. The Alternaria cornispora, this is the recent one, and the grey mildew, and bacterial leaf blight is there. And when you see the boll rot, which is the most emerging disease in recent years, which is along with the pink bollworm. The pest and this disease both are contributing for 90% yield loss in cotton of recent years. When I speak about the viral diseases, the 2 most important viral diseases, the cotton leaf curl virus and the tobacco streak virus. So when you see the disease scenario in India, the cotton is divided into north, central and south zone. When I speak about south zone, the TSP is more important in central and south zone. And when in north zone, the CLCV is the more problem, of which 90% yield loss was reported. So when I speak about necrosis disease, wherein it is caused by tobacco streak virus, so this is the most devastating one because it is a polyphagous nature. You can see 200 plant species has been infected, and this is first described in 1936 by Johnson. After that, you can see how it has infected. So the first is reported in sunflower, later by the groundnut in Andhra Pradesh. And also in 2003, you can see it has spread to the other crops, other horticulture crops also. And when you see the timeline, you can see 2005 only it is reported in cotton, that is in Warangal in Andhra Pradesh. And later that in 2016, it is the report in Tamil Nadu where you see 62% yield loss was there. And after that, the recent scenario for the past 5 years, it is keep on increasing in the southern zone of cotton cultivated places. So when you see, this is the virus of Ilar virus type genus of Bromoviridae family. and we have tripartite. So RNA 1, 2, 3 is there, and where 1 is responsible for its viral replication, and the 2 is for this RNA silencer suppressor, and 3 is for the movement protein and coat protein genes. So when you see in the field how these symptoms means, when you see in 45 days after the crop, you can see the first is the stunting, what it is given in the site. So this is stunting of the plants, first one, and the second one is the chlorosis and the necrosis, both together in the growing the young leaves. So after that you can see the necrotic lesions, and this lesion will spread to the petiole also, and the later stage you can see the drying of the squares. So this is where this is the drying of squares what we observed in the near 70 to 80 days of the crop. And when you see in arboreum also we have found this TSV. And when I speak about the barbadense, which is the Egyptian cotton, the symptoms are entirely different from the hirsutum, where we observe chlorosis and necrosis. Here we observe only the purple necrotic spot what I have given in the upper side of the picture. So, and you can also see this distortion of the leaf. This is the main symptom, and also the plant stunting. This is drying also the similar one. So why this slide I have kept means, when you go to the farmer's field, when you inquire about the farmers, they are unaware of whether it is means the deficiency or the viral infection. So the awareness is important. So this I have given that. The upper side you can see the extreme leaf reddening. This is the important problem in cotton. This is because of magnesium deficiency. The other is chimer effect, which is genetic mutation happens because of the chlorophyll pigment is lost in that place, particular place. This should not be confused with the chlorosis, which is caused by TSV. The other important symptom is the 2,4-D. When you have 2,4-D, it is extremely dangerous to the cotton. Even a small bit or a drift, it will causes the leaf initiation. You can see leathering of the leaf. So this is entirely different from our TSV. So we should know how to distinguish from the other abiotic stresses which is happening in cotton also. So when we speak as such, unlike the fungus or the bacteria, the virus cannot be cultured as it is an obligate parasite. Everybody we know. So we have to maintain our virus in a live plants. That is nothing but the artificial inoculation, wherein we used to create the aberration using the carborundum powder, which is silicon carbide. We have to make the cowpea, is the host, which is the local lesion host, which is for all virus we can use this type of cowpea plants, which is for sap transmission. So we have to use this phosphate buffer to inoculate it, and after that we can see the necrosis and chlorosis symptom. And this is the first assay, and after that we can continue to maintain the plant in the live, the cowpea or the cotton plant, or else we can go for the assay like molecular assay for RNA isolation and the RNA characterization, and also for the serological assays. So this is how we maintain the plant and the virus in the live plant. And the other, it is a vector transmitted. So as we know that it is a pollen assisted by thrips. So the second instar larvae of thrips is responsible for the vector transmission. So when you see this evidence of the pollen, of the Parthenium pollen, and the vector multiplication will make the severity of the disease in the field. So when you see the integrated management, this I will cover the later part. So when you see among all the management, when you speak, means the breeding for resistant varieties is important. Why? Because when the plant is tolerant to that particular disease, if for example for the TSV, then there is no escapism will be there. So for that we are going for the screening. Initial step for the developing of resistant variety is just screening. So when I take this project, it is in Barbadense. It is a major problem in South India where it is cultivated. So 300 germplasms we have screened for the 2 years, and this among the 300 germplasm, 14 are highly resistant and 94 highly susceptible germplasm lines were selected. And this is, since it is a breeding plot, I have taken the other study in a separate plot. So where we developed a novel standardized screening technology for this TSV in the field. So where we have to sow susceptible lines in 4 rows along with the resistant line, and along with we have to grow for a susceptible check, as a sowing is a susceptible check, and we have taken here to create the disease pressure and also the vector pressure population. We have maintained here the bendy, 1 row of bendy, chili and black gram. These are cultivated, and we have to go for the screening, and we have to observe for the disease occurrence at 45 days and also 60 days periodically. And we observed that this 162, ICB 162R, which is the among the 14 resistant line, this performs well with the less disease score and also for the virus. And this is the correlation heat map. What we developed is from the agronomical features. When you see that the TSV disease incidence, we can see that the number of sympodial branches and number of bolts per plant was positively and significantly correlated with the incidence. It speaks that when the disease incidence is less, these two features means the number of bolts or the sympodial branches will be reduced.
5 minutes
Yes,
more.
it's over, that's also. Then when you see, this is the important one, because why means, when you see in the field, here we maintain the Parthenium plant in vitro, because you can see this Parthenium plant as a reservoir. So during the off season, this thrips will breed in the Parthenium plants, and when cotton is cultivated, this thrips will transfer this virus through the pollen from the Parthenium plant to our cotton plant. So in this manner, it is multiplying and spreading the virus from the infected to the healthy one in the field. So this is the one more study. It is because how the day of difference in the sowing date affect this virus means for the consequent 2 years we have seen in Sweden and MRC, it is a hybrid ELS cotton. So we see that when the cotton is grown, and this particular Sweden is sown on 29th August means when the crop reaches 60 days, the disease incidence was more. So what I have to tell from here is, for the farmers, when you go for suine or the ELS cotton, they should take up the cultivation before the last week of August. They should take either by the 9th of July or the 4th of July in order to reduce the incidence. Why? Because during this 60 days, the crop will reach during the month of November. During the November month, the prolonged winter, this is susceptible for the barbitans. So along with the abiotic stress and our TSV, so we have severe square drying will be observed in the field. So when you talk about the epidemics, how it TSV contributes means based on the abundance of thrips population and because of mixed cropping like sunflower, groundnut together, sesame, everything. And also when you see TSV is it's a polyphagous nature. So it will also reduce the crop productivity also, and also it results in epidemic protection also. So when you see how in food safety means, it is a non-edible crop. Though it is a non-edible crop, when the cross infection of thrips will happen when it feeds the cotton and simultaneously when it feeds the sunflower or the other groundnut plant. In that way, it will result in the cross contamination. And also when you go for thrips management, we will be using the insecticide. So in that way, when you use more of insecticide, the pesticide also, it will make the environment unsafe for the beneficial insect population. That also we have to take care. And when you see as a food security means, where because of the low cotton seed yield, it indirectly contributes to the livestock feed. So in that way, the cyclists get affected because of this epidemic nature of the virus. So when you see this management means we have to go for vector control and also host resistance, what I have said, given the talk. And also we got to go for epidemic surveillance. What we are talking about is the spatial monitoring should be there properly at the early detection. And also we have to educate the farmer regarding the disease management, particularly integrated management, to motivate them to reduce the use of the insecticide, rather to go for the neem-based products for control of the pest, the thrips. So I thank the organizers of this seminar to give me an opportunity to deliver my talk, and also to the director of CPPS and head of the department, Dr. Angapan sir, and also our director and head for this opportunity to talk over a gathering. Thank you.
Thank you, Dr. Valamurthy. You presented in time. We will allow for 1 or 2 questions. Yeah, please.
Yes, I have a question. I'm here. Madame Yobino.
Yes, ma'am, I didn't see. Sorry, ma'am.
I just want, there are many viruses infecting cotton. So I wanted to know if, do you think that there could be any interaction between different viruses with TSV, or are you sure that the symptoms that you observed and cannot be also caused by an association with another virus?
But as of now, the 2 viruses are still reported in India. When you see cotton leaf curl, which is only in the north zone, Punjab, Haryana, Rajasthan, where it is cultivated, the cotton. So that is DNA virus. And when you see the central and south zone, this is the only RNA virus which is infecting in south zone. We are not seeing any interaction with the other viruses, ma'am. And we have one more virus also, cotton blue disease is also there, and cotton dark virus is also there. This is just reported, but it is not we are observing in the field. We are observing only this virus as of now, ma'am.
Okay, thank you.
Yes, ma'am. Thank you, ma'am.
Excuse me, Alarmadi.
Sir.
It's a nice presentation.
Thank you,
And
sir.
my question is also related to Buria questions. Why the leaf curl is severe in North India, whether these two, no, other TSV is severe in central and
Central and south
south
zone, yes, sir.
zone. Why?
Sir,
What is the main reason? Is it weather or?
So it is not because of weather, it is because of cropping system, sir. Here it is originated, sunflower only the first reported only, no, sir, TSV.
TSV okay.
Yes,
leaf
sir.
curl. Very,
Leaf curl, yes, so only it is present only in north zone only, sir, as far we have seen. We are not seen any symptoms since we are going for a periodical survey also. We are observing only in north zone, sir. Because of the whitefly dominance over the presence of whitefly is more, sir. Here we are not seeing that much of whitefly population here.
Now, is there any variation in the biotype of white flies in cotton?
Biotypes of whitefly, so I am not aware, sir. But as of now, we are not observing any symptoms of the CLCU in here, sir, in south zone and central zone, sir.
Thank you.
And similarly, we are not observing any TSV symptom in north zone. So they are distinguishing it themselves only CLCU in north zone and this is in the south zone, sir.
Is there any variation in the varietal distribution or hybrid
Yes, sir. Yes, sir.
among the regions?
Yes, sir. In hybrids only, we are reporting more of TSV, sir, compared to the varietal in cotton, sir.
Parthenium anther is having the virus. Have
So
you
this
proved that one?
this pollen from the latent infection pollens, no, sir, from Parthenium, we are just taking it that and we are inoculating it over the cotton leaves and also the copy leaves. Sir. There we are observing the symptoms, sir.
So you proved that the
Yes, sir.
pathogen
Yes,
is
sir.
from Parthenium.
Yes, sir. Parthenium. Sir, actually, as far as hirsutum hybrids, BT hybrids are more susceptible, sir. In RCH 629, and it is more, we observe near less than 45%, sir. Sir, hirsutum is fully affected, sir. Compared to barbadans, because barbadans only selected places we are growing in South India, in Karnataka and Tamil Nadu only. Which one, sir? Species means both are equally susceptible only, sir. Hirsutum and barbadans, sir. So arboreum, herbaceum we are not observing, no, sir. We are not cultivating also, just only for experimental plots we are maintaining. We are not observing anything. Yes, sir. In arboreum we are seeing, sir, but it's very rare, only observation, that occurrence, that's all. Not up to the infection and all, sir. Weeds also there, yes, sir. So weed management is the first step to control the virus, yes.
Okay, thank you, Dr. Valarmadi.
Thank you,
The rest
sir.
of the questions you can discuss with her during the 3 hours. We can invite the next speaker, Dr. Jean, on modeling for agroecological crop protection. Expect that by 1:20 you will be able to wind up. Now it's 1 o'clock. 20 minutes.
Okay.
Okay, so sorry for this technical matter. So a little bit of energy. It's the last talk before the break. Is it okay? Oh, because I do need the slides. It will be hard without the slides. Ah.
So, yes. Yes, do you have some animations in this? Okay. That's right. No. Yeah, if you don't mind, you take your own time to set it. We'll invite the other speakers, okay? Yeah. Yes. May I request Dr. Priyank? Sorry, you take your own time to set it and we'll, yeah. Next, Dr. Frederick, kindly keep your slides in the presentable form. If there is any problem, you kindly sort it now itself. Where is Dr. Frederick? Yeah. You keep ready your slides with your slides after this presentation. Okay.
Good afternoon to all, and thank you, sir, for the surprise. I was thinking my number will come after lunch. Yeah. So actually, today I am here to present on pest and diseases of potato and its management in Nilgiris. So basically, I am Dr. Priyank. I am working as a senior scientist in Central Potato Research Institute, which is located, actually our headquarters is located at Shimla. We have 7 regional stations, one is located in Ooty. So I will directly hit to the point. So about potato, everyone knows it is important crop, third major crop after wheat and rice, and then India ranks second after China. Even China is having more production, but productivity wise India is at top. We have almost double production than productivity than China. Coming to India, we are growing this in India. Potato is being grown in plains, plateau and hills. However, Nilgiris are the only place where this potato can be grown throughout the year. If you ask any South Indian, they will prefer, there are two, they knows only 2 potatoes. One is Ooty potatoes, another one is North Indian potatoes. And always the price for Ooty potatoes is on almost on top because of the taste. Okay, why that taste is getting developed? Because of the throughout around the cultivation is possible. Throughout the year we have 3 different seasons in Nilgiris, and because of that this particular taste, they are getting very good taste. So actually this round year cultivation is possible. Actually this is the only place in world we can say where 3 crops of potatoes are possible in India, that is in Nilgiris. So actually this is because of suitable climatic conditions and that particular place. Okay, so when there is a suitable climatic conditions and availability of continuous availability of host, actually there is some disturbance in the slides, sir. So okay, when there is continuous availability of host and suitable climate, there is continuous, we are expecting so many pests and diseases. New pests are emerging. There are some several old pests, they are flourishing, and new pests are also coming. So among that actually we can categorize this particular pest and diseases in 2 major categories. One is major and one is emerging. So coming to major, actually we have 3 major pest diseases over there. First one is, first and foremost, it is lead blight, followed by potato cyst nematode, and then third one is serpentine leaf miner. However, there are, in these recent years, we are experiencing the problem of potato tuber moth in storages, bacterial wilt and common scab of potato. So coming to lead blight, everyone, Andy sir was talking about this Irish famine, which has experienced in Ireland and in somewhere in 1845 to 53. So almost 1/3 of the population of Ireland died, 1/3 moved out of Ireland, and Ireland was left over only with 1/3 population. You can see this is the photo I used to always showcase. So this is our trial plot with 3 by 3 meter square plot, and then this particular plot was not sprayed or used with any chemical. And then when at harvest, we can see out of 2.5 kgs we have harvested around 25 to 30 kgs here. However, in this plot hardly 800 to 900 grams small tubers. So whatever we are planting, that much also we cannot harvest. So this is the condition, and Ooty climatic conditions are too harsh. Always the temperatures are below 20 and humidity is above 90. So in that case, those are the most favorable conditions for lead blight to develop. So actually we can say 90, I am telling more than 95 to 98% losses we will experience if we don't have any option. So actually how to manage this particular pest. So this is the, this particular slide shows how the disease develops. Once we are getting, first we will get the disease on the leaves, and then it will come to then stem and then entire plot. This particular process takes hardly 7 to 8 days. Okay, after that entire crop will be black if you are not taking any measures. So actually how to manage, people are mostly dependent on susceptible variety, because we have certain varieties which are resistant to lead blight, that is Kufri Girdhari. I will show some of the slides. Kufri Girdhari is one of the best varieties suited for Nilgiris, and but the problem is the availability. Ours is the only station who are involved in production of seed of these particular resistant varieties, and along with our state department, we are producing hardly 200 tons. However, we require almost 6,000 tons for to meeting out the requirement of Nilgiris. So actually, people maximum, because this particular Kufri Jyoti was released somewhere in 1968. So it is completely in our seed chain. So easily that's the seeds of Kufri Jyoti are getting, farmers are getting those seeds. So actually what people are doing, you'll not imagine, if you are very happy to eat Ooty potatoes. But if you are seeing Ooty potatoes plus chemical residues, you will get at least 10 first links, okay, that shows that in Google, Ooty potatoes are highly it is contaminated with one or other pesticides. And actually what we are saying, minimum farmers are using 15 to 20 number of sprays in a crop season, that is 90 days. Every after 4 to 5 days, they will go for one spray. When they are seeing the sky is clear, they will enter with sprays. So that is the problem. So actually how to manage this particular issue? So CPRI has developed one forecasting model that is called Indo Blightcast. And then this particular model is very simple. Here it requires only maximum and minimum temperature and humidity. That we have to update, and then it will give us the predicting of this one late blight in 3 different colors. Green means no need to enter in the field, because this is the weather conditions are not suitable. So many times this is like farmers, they were not aware when to enter in the field and when to stop the sprays. Yellow means the weather conditions are getting suitable. However, red means now you have to enter in the field. There is no other option. Anytime late blight will appear. So actually we are doing continuously this particular modeling in our place. This is Pan India model. Because of this, actually our CPRI team, they got Netaji award for our Nanaji Deshmukh award, team award. Okay, and then we can see here in 2021, 2022, actually I kept different colors, but here some disturbance is there. No issues. Actually we are using this model for prediction as well as for scheduling the fungicides. And then farmers practice is minimum 14 to 15 sprays. However, we have, because of this particular model in 2021, in our seed crop, we have sprayed only 8. However, in 2022, the condition 6-axis, this is the red color. We can say actually red color, red days. Actually, I told in Ooty always we will get more humidity. So more humidity in 2022, the red days were more. Okay, comparing to 2021. So in summer season of 2021, we have administered only 8 sprays versus 10 sprays in 2022. So almost 7 to 8 sprays we are saving comparing to farmers' practice. The same thing they have used in Uttarakhand and West Bengal, and because of this model, more than 2 lakhs kgs of one chemical has been saved just by giving one advisory. Please don't go for spray because the conditions are not suitable. So that much chemicals we can save with this kind of models. So students are here, they can work with this kind of things. Okay, really it will be useful for the farmers. So actually in continuation we are evaluating new molecules which are in the market. And then because farmers, if you are going to agro shop, whatever we are suggesting, the agro shops will suggest the double of the time. Okay, so actually we have one story I used to tell in my all the lectures. Actually, we have one village in Adhikaratti. That is one of the biggest village of Nilgiris. We have one village. And then you will not believe, maximum people in Nilgiris are Badgas. So Badgar is one of the community. Major food of Badgas are generally potatoes, beans and beetroot. And you will not believe, in single village we have more than 35 breast cancer cases. Morning, sir was talking about, we have to accept, we are responsible and we have to do some extension so that the pesticide usage can be reduced. This is one of the way by modeling Indo Blightcast. Like that, some of the tools we have to come up with. And then we are doing, and then we are recommending farmers what are the things we have to go for. Coming to, I was talking about resistant variety. You can see this is Kufri variety, Kufri Girdhari. This is our farmer. We used to take on-farm trials. And then this farmer is from Kotagiri. Almost at 90 days, this is the 90 days crop, almost summer season. Summer means one of the susceptible season for late blight. And he has not taken a single spray. So like that we are doing so many, this one. We have one more area like Munnar, that is in Kerala. So like Ooty, we have one more replication of that climate, that is in Kerala. So we used to give 100 kgs of each variety. Jyoti, susceptible, Himalini, moderate, and Girdhari, resistant, highly resistant. With one request to the farmer, please don't spray. If they are not spraying, this is the result. Out of 100 kgs, hardly 20-30 will be coming. Out of 100, hardly 50 to 60 will be coming. However, 3-4 bags are there. So out of 100, 400 to 500. Last day we will be organizing one field day or farmers day, and after seeing, the farmers will ask next time we want this particular variety. Like that, actually one of the farmer, he came, he was about to stop potato cultivation, but he's from OT, and then he has taken 300 kilos from us. You will not believe, till now he has sold more than 30 to 35 tons of seed potatoes to the farmers, okay, nearby farmers. So our duty is to convince only one farmer, he will convince another 100 farmers. So this is what we are doing on farm trials, that is a regular activity over there. So, but still we are suggesting avoid growing a single variety on the same piece of land. Why? That I will show you later. Coming to the next most important problem, that is PCN. So actually PCN, I'll be very happy to listen Dr. Professor Hamlin. So our this is like you know, our both are working on the same aspect. So we'll be seeing what he will be dealing with. I'm very happy to see that particular concept what he will be presenting. So he is Dr. F.G.W. Jones. He was from Rotherham State University, and he was on his personal tour to Ooty in 1961. So up to that time, nematology was not a separate science in India. So he came and he reported first timely Globodera rostokensis from India. Okay, many of the students, please note down, these are the points. 1961, first report of Globodera rostokensis from India. 1963, ICR has launched golden nematode scheme. Under that, large-scale inspection was carried out, and they found that PCN was restricted only in the Nilgiris. So they implemented quarantine in 1971, and in 1971 to 75, there was one Indo-German Nilgiri development project. Under that, Germany government has given one chemical free of cost. So actually, history of chemicals is there from long. Okay, almost they have dumped almost 10 lakhs kgs of this particular chemical in Nilgiris, and after that they surveyed, but the trends were not promising. So you can see the area of potato came down. The nematode population I have given here, but the things are missed now. So those actually wherever the population was low and medium, it has reached to high and very high in all 4 taluks of Nilgiris where potatoes is being grown. So actually people from our station, they have started breeding program that is under the name of SON, Studies on Nematode. Okay, and then these are the 2 species what we have, that is Globodera rostokensis with yellow females, Globodera pallida with white females. So 2 species we have different with the different pathotypes. This is how one single patch it will spread to entire field. But when we have to confirm, you can see only confirmation is presence of white or yellow females on the roots at 55 to 60 days. This is how we have to confirm it. So if I ask our audience how much losses nematodes are responsible for, but actually the answer comes hardly 10 to 15% because we can't see them by naked eyes. Pathogens, yes, we can see, definitely people will accept pathogens are responsible, pests are responsible. What about nematodes? So actually this is one of the story from our end. So we have 2 varieties I mentioned, one is Kufri Girdhari, another one was Kufri Swarna. So Kufri Swarna, I told SON was the project Studies on Nematode. Under that, in 1985 they have released India's first nematode resistant variety, that is Kufri Swarna. Okay, in India if we say potato research, it started in 1935. Our Swaminathan sir was the first to work on potato breeding also. And in 1985 we have released this variety. That year was the golden jubilee year for potato research in India. That's why this particular Kufri Swarna. Swarna means gold. So that's why they have given this Kufri Swarna. So we have one variety, we can say this particular side, white flowers. So this is Kufri Swarna. The same plot, here we have planted Kufri Girdhari. In 2014, by harvesting this, we have earned around 139 tons of potatoes we have harvested. And we used to sell it as a seeds. So out of selling, we have earned around 28 lakhs. But farmers, even our scientists never appreciate nematodes. So farmers, they requested joint director, we want only Kufri Girdhari because we are having this late blight as a major issue. And joint director met our director. Director passed that information to us. We have to grow only Kufri Girdhari. And in 1 year, you can see the results. 139 tons and 139 tons, we got only 60 tons. However, where we earned 28 lakhs, next year it was 8 lakhs. More than 56% reduction in terms of yields, more than 70% reduction in terms of monetary return. This is what nematode can cause in a year if the populations are high. So actually, that's why I told that tagline ki don't grow a single variety on a same piece of land year after year. Even we have to alter the varieties as well. Okay, so when we have 3 seasons, so first season the late blight will be less, so you grow resistant varieties to nematode. However, next 2 seasons you can grow the resistant varieties to late blight, like that. So actually, how to manage this PCN? So seed we have to use healthy seeds. We have developed one seed treatment where if your seeds are major carrier for cyst nematodes. So if you are treating seeds with 2% sodium hypochlorite for 30 minutes, it will decompose. It will just destroy all the cyst nematodes. So you can use those seeds. Then actually we have several crop rotation, organic amendments, biological control. I will concentrate on resistant varieties. So I told SON 76110. 76 was the year and 110 was the hybrid number. And then that variety was released in 1985. Followed by we have released one more variety in 2020, that is 2019, that is Kufri Sahyadri. So both are having excellent, this one resistant to nematode. 1985 release variety, till date it is ruling in OT. 40 years today it is completing. So that is the achievement. That is because of crucial screening what they have done, standardized during early period. Okay. So actually this is the single plot. This is Sahyadri, this is Jyoti. Because of non-resistance to late blight and potato cyst nematode, it has vanished by around 60 days. Versus you can see this is not cropped photo or something, same field, one single photo. Okay. And then this Kufri Jyoti in Sahyadri, if you have any rogue plant, sometime it will be escaped. But if the plant is not having resistance, you can see the difference. Okay. So actually this is the side of, I told, Kufri Girdhari. This side Kufri Girdhari. At 60 days after planting, this will be the condition if the PCN populations are high. So actually what we are suggesting the farmers, don't grow Kufri, any variety on the same piece of land year after year. These are actually, this variety like Kufri Girdhari, we are popularizing this one also. This is one of the farmers from organic growers. And then in organic cultivation, it's very difficult to get rid of nematodes. So our technology, they are using in spring season. This particular person, he is doing in staggered way, so that he can harvest maximum in next 2 seasons also. So actually like that we are doing, we have one more technology called trap crop. So there are 2 types of trap crop. One is susceptible trap crop followed by resistant. In susceptible, the harvesting at right time is crucial. Otherwise you are going to add the nematode culture in the soil. But we have the other part, that is resistant trap crop. Solanum sesamrifolium is one of the most preferred one in European country. And then there is here no need to worry, because earlier plant will mimic the potato roots, so that nematodes will hatch out, they will enter, but they are unable to produce males and females. So actually this is how it works. In potato cyst, after sensing the root diffusates, the nematodes will hatch, they will enter, and they will develop into male and female. Males will come out, fertilize the females, and it will start laying the eggs. However, in case of sesamrifolium, because of root diffusates, hatching will take place. They will enter also, but further development will not be there in the roots. So by that, actually, we can get almost more than 90 to 95% reduction in nematode population, soil population. So what we are suggesting, one spring season you grow Kufri solanum sesamrifolium. In next 2 seasons you can grow any variety. Now actually, again, one particular strategy will not work for any pest and diseases. So we have to integrate different strategies. Coming to 2020, we were enjoying quarantine, and then farmers started calling us for serpentine leaf miner. Yes, sir.
Yes,
okay.
please.
So 2020 actually this happened. This is how actually that symptomology it started. First, this particular fly used to scrap the leaves. Then they will lay the eggs. After that, you can see from this particular single leaf, I could identify almost more than 21 maggots of this leaf miner. And then it will devastate entire field. So initial stage we have to apply this particular yellow sticky trap. That is one of the, this is heavily infested field. We can see this is 75 days old crop. In that year, farmers have experienced more than 40% losses in Nilgiris. Okay, and then because of our interventions, we have identified that particular variety, and that was the first report from India. No one was knowing how to control that. We have given some of the management practices, including cleaner cultivation, because these are the major crops of Nilgiris. In all the crops, the infestation was there. However, in wheats also infestation was there. So what we have given, cleaner cultivation, followed by we have to apply yellow sticky trap at right time. That is 30 to 35 days after planting. We have to use abamectin, because abamectin is one of the chemical where that is accepted in organic cultivation as well. And we have to make aware farmers how to manage this particular pest. By this we can, now the infestation has been came down below 4%. Farmers are not facing any problem in potato. Now coming to the emerging problems, this is PTM, one of the major issues in storages. So actually you can see it will infect field as well as in the stores. So actually there are certain management practices which are being used since long. We should not keep it like this. We will learn in our books, like we should not keep the potatoes dug out in the field by evening like that. Okay, but
Another
actually
2
we
minutes.
we have developed actually this particular, we have identified one EPN and then that entomopathogenic nematode. So this nematode is killing the potato tuber moth. So actually we have studied that effect. It is working well. Now we are working on formulating that particular nematode. Additionally, we have developed one physical trap, and then recently we got patent for it. And then actually this particular trap we have tested against, we have used these pheromones as one of the control treatment. We got these pheromones from Spain. That is the only technology available for PTM throughout the world. Okay, and then our trap, I can say proudly, those pheromones trap, we could get around 63 to 64 males by 7 days. However, our trap, it has, you can see this is the single day catch, more than 400 PTM adults in a single day, single night. So actually this technology we will be commercializing soon, and it will be farmers friendly technology. Hardly with 2,000 rupees it will be available for the farmers. So that is what our aim is now. Now actually in addition to this PTM, we are actually experiencing this year bacterial wilt as well as common scab of potato. So here we need to work with the TNU. Is there any, if any person or any student is interested, please come ahead, because we are experiencing huge problem in Nilgiris now. That's it, last slide, sir. So actually I would like to summarize my presentation in one small time infograph. So you can see in 1950s, potato become major crop and late blight became major disease. In 1957, our institute has started in Ooty. In 1961, PCN was reported by Jones. 71, quarantine was implemented. 1985, Swarna was released, which was the result of that quarantine, followed by SON, studies on nematode. In 2019, we have released one more variety, Kufri Sahyadri. Both are ruling now. In 2020, leaf miner came. We have given the solution for that. However, we got one project from, we are working with one firm, that is Green Signal Biopharma Company from Chennai-based firm, and then they are the maximum producer of BCG vaccine. So when they used to take these potatoes from market, they actually, that is one bacterium they are using, that is Mycobacterium bovis. So that particular bacterium they are cultivating in potato. Okay, and then what we are getting, our children are getting, that is BCG vaccine. So actually when they are using they were using the potatoes from the market, they never used to get that good yield. And then they have contacted us, and then past 1 year we are providing them chemical free potatoes. And then they are producing this BCG, and we are very much proud to say that now in India or in WHO, they are major providers to WHO also. So actually whatever BCG vaccine is coming, that is from our end, and then we are very happy to share this with you all. Okay, and then they want to continue this one for another 15 to 20 years. Okay, so this is the thing. So finally, it is important crop. These are the major problems: late blight, PCN, and leaf miner. Resistant varieties are options, and then integrated management we have to follow. In future, yeah, definitely. Morning also people were telling about AI technologies, AI tools. We have started actually recently, last week only, we have signed one MOU with SSN College of Engineering, and then we'll be developing some of the tools for early detection and management of the pest and diseases of potato, and some newer and fewer options we have to explore. So with this, I would like to thanks all the organizers of this particular conference for giving me this opportunity, and all the audience for patience listening. Thank you.
I think we can interact during lunch hour with Dr. Preen if everybody agrees.
Okay.
Because we have to accommodate 2 more presentations before lunch. Or you can have quick questions, 2 questions.
I think everyone came prepared with 30 minutes, but
No, morning itself they told 20 minutes.
Yeah.
Okay, thank you very much for the nice presentation. I
Thank you,
have
sir.
two questions. One is related to the forecast system,
Okay.
related to lead blight, because in each European countries,
Yeah.
like in India, farmers use decision support system to help trigger fungicide application. And I was wondering, do you have any measurement of the quality of prediction of the underlying model? And what would be the value in terms of any statistics?
Yeah.
And the second question is, do you have any data set that links the dynamics of symptoms of late blight to yield losses? Because a couple of years ago, we designed a yield loss model that worked very well from American cultivars, and that worked greatly for 8 French cultivars. And I'm curious to see if you have that data set, we could try this model on your data and see if it works or not.
I think Dr. Priyank, you received the question.
Yeah.
With your permission, you kindly interact with you. You explain.
Okay. Right.
Okay. We will invite the next speaker.
Okay, thank you, sir. Yeah, thank you.
Hello everybody, so today I'll talk about durable suppression of Globodera pallida, the pale cyst nematode, bioresistance, genetic resistance, rotations, and biocontrol. So as I mentioned before, I'm a mathematical modeler, so I'll present a model which is quite complementary to what our colleague just presented before. So first, let me present myself shortly. So I got a PhD in mathematical biology, so I'm not a biologist like most of you. And now I'm professor of quantitative plant disease epidemiology in the Institute of Genetics, Environment and Plant Protection, which is our research institute in Rennes, France. So this is in Brittany region. You can see here a picture. Okay, so I'll talk about potato cyst nematodes again, maybe more, maybe slowlier, and how they can be controlled in France, in Europe, and then I'll show you some elements of mathematical models to get some intuition about how we can control these PCNs, and then I'll present results and the main messages. So potato cyst nematodes. So you can barely see them. They are microscopic round worms and nematodes in general. Sorry, and the nematodes are the most abundant animal on Earth, including in the oceans. And some nematodes, so they benefit the soil, but other nematodes, as we've just seen, they infest crops and soil. And in particular, some kind of nematodes, so the potato cyst nematodes, they infest potato, as we've just seen, and this is the world's fourth largest food crop. So it's cultivated everywhere in the world, in America, of course, also in Europe, and as we've just seen in India, for instance. So the symptoms they cause are growth retardation and early senescence, which results in between 20 and 80% yield losses in potato fields. So economically, the cost due to these yield losses and also the control measures reach billions of US dollars per year worldwide. And so these piston species, they are present on quarantine list both in the North America and in Europe and maybe also in India. So this is just to say that this is quite a big deal, this nematode. So you can see here a picture from INRAE. So this is the root system of a potato, and the white circles that you see are the cyst of the nematode. So there are many cysts on the root system of this plant. Okay, so a few words about how we control PCNs. So for decades before, pesticides, more specifically nematicides, were used to control these nematode species. So for instance, by injecting nematicides, so these products in the soil via these kinds of machines. And the problem is, as you know, that the pesticides inflict collateral damage on other life, including humans, as we've just seen. And so over the past decade, at least in Europe, the most efficient nematicide have been completely banned. So that means that the growers, the potato grower, they cannot use those methods any longer. So we have to find solution for them. So they need other control methods, and genetic resistance is a promising alternative to nematicides. And they can be very effective for PCN control. The problem is, as often in plant protection, resistance breaking nematodes or some mutants have been found in Europe. And so we fear that they will invade if we use only genetically resistant cultivars in the field. So we have to use these cultivars with parsimony, and maybe in combination with. So other control methods includes the rotations in time, as we've just seen, and biocontrol. So I'll tell you what I mean by biocontrol in a minute. So this is a picture again about this cyst of the nematodes. So the cyst is actually this is the female nematode, which will become a cyst, and which is, maybe you can see here, which is fecundated by a male, which is the ring that you can see here. Yeah. So just a few words about the PCN life cycle. So their survival form are the cyst, and so each cyst can contain hundreds of eggs. And so when they get into contact with root exudates, which are molecules that are produced by the roots, these cysts hatch, and so they release larvae of stages, and these larvae can migrate and infect or infest and penetrate into the root system of the host, and then when they are in the root, then they grow, they use the resources of the plant, of course, and at some point they differentiate either into a male or into a female. And so everything going well if the plant is susceptible, so the pathogen, the parasite, produces like 40% male and 60% female. And then these males can migrate outside of the root while the female remains attached to the root. And so there is sexual reproduction, and then the cyst contains the eggs, and we start a new cycle again. I will not go into detail into this talk, lack of time, but an interesting thing is that genetically resistant hosts, they can just they proceed through masculinization. So that means that the larvae of the nematode, they go into the root, they grow, but somehow they sense that the plant is not a good resource for them, the resistant plant, and so they produce only males that will migrate, and they do not produce females. And only females, of course, produce eggs, which will be the survival forms of the nematodes. Yeah, so I promised that I will tell you what I meant by biocontrol. So this is again the life cycle of the nematode when everything is normal for the nematode. And so again, the root system of the plant emits root exudates. And an interesting thing is the system is that this molecule, these exudates are very specific to Globodebra pallida. That's very specific nematode species. So there are very specific molecular interactions between the root system of the plant and the nematode species. And yeah, when they get this signal, then they hatch and they produce this larvae, and the larvae then infect the host. So that means that if we make rotations with other host species, then this rotation may not be that efficient, because the cysts, they can survive very well for years or if not decades in the soil, and they just wait for the signal to hatch and release larvae. So that's a problem with respect to rotation, this high specificity. And and so the trick here is has been developed by researchers at my research institute, nematologists, in collaboration with a private company working on the production of seed potato. And so they succeeded to produce the same root exudates, and to show that one can use this root exudate to make the cyst hatch in the soil. No, thank you. And to make the cyst hatch in the soil. And so in the absence of host plant, so that means that the cyst just die in that case. And so the idea is to apply these molecules in between the cropping seasons to kill the nematodes. Okay, so the idea is to combine these three possible control methods. So resistance, genetic resistance by planting a resistant plant, of course, rotations and biocontrol. So on year one, we can plant a resistant plant. On year two, no resistant plant, no potato plant, sorry, something else. Same thing on year three, same thing on year four. So these are the rotations. So that means planting one potato every 5 years. And the idea is to apply these fake root exudates each year where we plant something else than a potato. And the question was whether it could work. So the problem with this kind of system is that, as you guessed, it's very difficult to make experimental tests of this strategy. So the combinatorics of the possible strategies, the number of years for rotations and so on, can be quite high, and the experiments would be very long and would maybe require some space. So the idea is to use models first to reduce the dimension of the problem and test more specific combinations of the controls in the lab then. Yeah, so it will be too long and costly, and so the idea is to use mathematical and computers to test scenarios through multiple years and these strategies. Yeah, so what I'm going to present very briefly is a so-called demogenetic model. So that means that the model keeps track of both the genetics of the nematode species, whether they are avirulent or virulent, and also the population density of the nematode species. Oops, I did something bad. I cannot go forward. No, I can. Okay, sorry. My bad. Yeah. Okay, so in that kind of model, we use mathematical notations. So this is a short description of the model. So these are the larvae of the nematodes, which may, the juveniles may differentiate either into male or into female. And then there is reproduction, and so the cysts contain eggs, and they survive, and so on. So the model can be quite complicated. So just to give you an idea of the kind of model I use, I presented the simplest possible version of the model. So meaning in a susceptible host population, we have a recurrence equation that links the nematode population density N on year T with the nematode population density the year after. So this is this is this application. So what you can see here is the time, the number of nematode generations. So the nematode, they do only, in France, they do only one generation per year. I have a question in India, but we'll see later. And so, yeah, this is time in year and the nematode population density. And so with this equation, the population grows logistically. Yeah, and there are two parameters. K, which is the nematode carrying capacity, so the maximum number of nematodes per gram of soil. And R is the reproductive number of the nematodes, so it's a common parameter in epidemiology. So, yeah. And an additional parameter that is important is the male fraction of the nematode in the progeny in a susceptible host population. Yeah, so that last parameter, M, the male fraction, is relatively well known. It's 40%, so we have an idea of the value for this parameter. What was more tricky to obtain was an estimation of the reproductive number of the nematode. So this parameter, in fact, is the product of several processes, and we estimate that on a susceptible host, in the absence of any control, it can attain values like 54, which is just huge, epidemiologically speaking. So that's why we have so much problems controlling nematodes with susceptible varieties. Yeah, and so this is why we use resistant varieties and again biocontrol and rotations. So there are two additional control parameters so that I'm going to talk about. The biocontrol efficacy, B, which is the fraction of nematodes in the soil that are killed by the fake root exudates, the synthetic root exudates. And the second parameter that we can control is the rotation length. So this is capital L, which is 3 years here, meaning that we plant one potato every 4 years. Okay, so I go directly to the output of the model. So on the x-axis, we have the biocontrol efficacy, so the fraction of nematodes that are killed by biocontrol in the soil. And the y-axis, we have the rotation length. So the gray region is economically non-viable. So speaking with the growers, they say, okay, we may grow one potato every 3 or 4 years, but not more. So that it's not acceptable for them. And so we wonder what was the minimum biocontrol efficacy in order to get rid of the nematode in the long run for acceptable rotation length. So if we consider one potato every 5 years, so that means a rotation length of 4 years, then biocontrol efficacy should be at least 40%. So that means that, yeah, and there is like a bifurcation, so if it's lower than 40%, then you will not get rid of the nematodes in the long run, and if it's more than 40%, according to the model, that there is, the model says that we will get rid of the nematodes in the long run. So that means that biocontrol has to be efficient, but not incredibly efficient either. So it's quite promising. And so the next question is, okay, but how long will this take? And so we can make some simulation. I just present one simulation here, which represents the nematode population density as a function of the number of years or seasons that elapsed. And so with 50% biocontrol efficacy, we start with a relatively highly infested field on year 0, and then the nematode population density drops, and we see that during the rotation, so there was no potato during the first 5 years, and then we introduce potato again, and so the nematode population density rises again, and then declines, and then dumps. And so we can say that roughly after like 10 years, we might get rid of the nematodes according to the model. And what I mean here is that the density of nematode will be lower than 1 nematode per gram of soil, and that's a threshold in France, in Europe, at least in France, above which one's considered that the field is infested. And so that means that if your field contains more than, is tested and diagnosed and contains more than 1 nematode per gram of soil, then you're under quarantine, you cannot produce potato any longer. So that's the target here to attain. Okay.
So 5 minutes more.
Yeah, thank you. So preliminary conclusions. So resistance alone may not be sufficient to suppress the nematodes. Rotations are necessary but may not be sufficient either. And biocontrol, however, if it's used in combination with the two others, could make it possible to suppress nematodes. So that's encouraging for the growers and for us to develop alternative to nematicides. So in the last 2 minutes, I would like to mention that this model is very simple, so it's deterministic, so there is no consideration of genetic drift, for instance, while it's well known by my co-authors, my nematologist colleagues, that genetic drift is very important for this nematode species. So we wanted to go further and to include that into the model. So mathematically, that means creating a stochastic version of the model. So I will not present the equations, but basically the idea is that we put probabilities at each stage of the life cycle of the nematode, and we make many simulations to get some statistics about the nematode population dynamics. Yeah, and so typically this is what happens when you use a resistant variety. So you start from a highly infested field, and then the population of nematodes drops again. And then in some trajectories will go extinct. So that means that some of the nematodes will go extinct, but some won't go extinct necessarily. And so the virulent nematodes that are present in the population, they may survive and reproduce, and the population may recover thanks to these virulent nematodes. And there is a huge variability. So what you can see is the interval of confidence. So there is a huge variability in the nematode population dynamics. So there is selection for virulence when you use resistant plants, of course. And my point here is that approximately half of the nematode population that we simulated went extinct. So the probability when you do this that you just get rid of the nematode by extinction is 1/2, which is quite high. And so you can wonder what's the probability of the nematode extinction as a function of the initial frequency of virulence in the field. And so what this graph shows is that it decreases sharply as a function of the initial frequency of virulence. And so, what this means is that the growers should not wait to have like more than 20% of virulent nematodes in their field to use a resistant variety, because otherwise the probability of suppression will be less than 40%. They should do that very early when the initial frequency of virulence in the field is low to expect getting rid of the nematodes. And this graph is the same, but with the initial population size of the nematodes on the y-axis. So what this says is that the probability of extinction mainly depends on the initial frequency of virulence in the field, not on the population density. So these extinctions are mainly due to genetic drift. Yeah, and so I'm just about to finish. So the message here is that, using these 3 control methods alternatively to nematicides might be possible might be a solution. So what I presented just before in the deterministic model is actually a worst case scenario, because when you consider genetic drift and stochastic phenomena, then you have a possibility of nematode extinction. And interestingly, the probability of nematode extinction can be relatively large if the initial frequency of virulence is narrow enough. So the message is that growers should survey, so should test for virulence in their fields and control fast then. And the message for researchers, for nematologists, is that it should develop molecular tools to be able to evaluate the frequency of virulence in the field. And this is research under progress. Yeah, so I think I'm about to finish, but yeah. So I'd like to thank my collaborator, Israel Tankam, who is a postdoc who worked with myself and my nematologist colleagues, Jocelyn Montari and Sylvain Fournet. And if you're interested in this research, part of it has been published in this paper. Well, thank you very much. I'm happy to take any questions.
Thank you.
It's a very nice talk. I really enjoyed. Only one observation. You mean to say resistance is not a solution for nematode management. Is it because resistance breaking down or something else?
Yeah, it is due to resistance breaking down. Yeah.
So how frequently this is happening?
I mean, in plant diseases in general, this is quite frequent. That when people use genetically resistant variety with major resistances, meaning that block the infection against aberrant pathogens, then quite often, very often, after a few years, variant pathogens appear and break the resistance, while it takes like 1 decade for breeders to introduce a new resistance. So in that specific case, we don't know because we have only one data point, right? But what we know is that we have already variant nematodes that are in Europe, in the Netherlands, in Germany, and so if we just dumbly use the resistant variety everywhere homogeneously, for sure it will be broken very fast.
Okay, so mean to say that the new pathotype is evolving?
Yeah.
So the word extinction, what do you mean? Is the old pathotype extinction and the new one is emerging, or it is altogether the entire species is looking forward to their extinction?
In that model, it was the whole nematode population that got extinct.
Okay.
Yeah, because
the new pathotype evolves.
Both hibernant, so the older pathotype and the new pathotype, they go extinct together by using genetic resistance, rotations, and biocontrol.
So you mean to say the biocontrol option can delay the emergence of new pathotypes?
That's a good question. We did not really address this in that work, but that would be interesting.
This is a new line of thinking.
To explore. Yeah.
To delay the pathotype evolution, we can use other options like biocontrol, microbiomes. All those things are emerging now.
Exactly.
Thank you. It's a nice talk. Excellent.
Thank you.
Okay, thank you, Dr. Patrick, and I think we will close for this session for lunch because already we have taken half an hour extra till lunch hour. So with the organizers from information, we will. That's okay, fine, but people are hungry also. Thank you so much. I think we will after lunch we will meet. What time we should